Today we are going to talk about DNA replication in a prokaryote. In DNA replication, there are three main steps: initiation, unwinding, and elongation. There are several key terms to pay attention to as we go through these steps. Initiation is the start of DNA replication. An initiator protein binds to the origin of replication, causing local unwinding and a short stretch of single-stranded DNA. DNA gyrase is a topoisomerase located ahead of the replication fork. Its job is to reduce the torsional strain created by the unwinding of DNA by helicase. It does this by relaxing positive supercoils that would otherwise form during the unwinding of DNA. Helicase is located at the replication fork. It unwinds and separates the double-stranded DNA by breaking the hydrogen bonds between base pairs. This occurs at specific regions known as the origins of replication, creating a replication fork of two strands running in antiparallel directions. SSB proteins bind to the DNA strands after they have been separated. Their job is to protect, stabilize, and prevent double-stranded DNA from forming. These proteins also help to prevent the single-stranded DNA from being digested by nuclease. SSB proteins will be dislodged from the strand when a new complementary strand is synthesized by DNA polymerase III. DNA primase generates a short RNA primer of about 10 to 15 nucleotides on each of the template strands. The RNA primer provides an initiation point for DNA polymerase III, which can extend the nucleotide chain, but it cannot start one. Free nucleotides align opposite their complementary base pairs. DNA polymerase III attaches to the 3' end of the primer and covalently joins the free nucleotides together in a 5' to 3' direction. As DNA strands are antiparallel, DNA polymerase III moves in opposite directions on the two strands. On the leading strand, DNA polymerase III is moving towards the replication fork and can synthesize continuously. On the lagging strand, DNA polymerase III is moving away from the replication fork and synthesizes in pieces. These short fragments are known as Izaki fragments. As the lagging strand is synthesized, it has multiple RNA primers along its length. DNA polymerase I removes the RNA primers from the lagging strand and replaces them with DNA nucleotides. Finally, DNA ligase joins the Izaki fragments together to form a continuous strand. It does this by covalently joining the sugar phosphate backbones together with a phosphodiester bond. If DNA polymerase I is absent or stops working, the RNA primers will remain on the lagging strand, possibly leading to a mutation in the DNA sequence. However, if the mechanisms within the cell pick up on the mistaken DNA replication, the cell cycle will possibly not continue and lysis will occur. Thank you so much for watching.